Hello everyone. So in last video lecture, I talked about biotechnology. So in this video lecture, I'll talk about fermentation. So first, let's start with the definition. So what is fermentation? So usually, I mean, there are some conventional definition, like for example, Pasteur's definition. So like conversion of sugar to alcohol using yeast, chemical conversion of carbohydrates into alcohols or acids, any spoilage of food by microorganisms, any process that produces alcoholic beverages or acidic dairy products, or we can define it as any energy releasing metabolic process that takes place only under anaerobic conditions. These all are actually regarded as definitions of fermentations. However, these are kind of more general type definition. Usually, biochemist and microbiologist, they have different meaning for fermentation. Okay, so they define it definitely. So in case of biochemist, they means fermentation this way. So biochemical meaning relates to the generation of energy by the catabolism of organic compounds. Okay, and whereas uh, in microbial cells, fermentation may be defined as process of growing of a culture of microorganisms in a nutrient medium and thereby converting feed into a desired product. Okay, however, so in general, or in a simplified way, we can define fermentation as the process in which a substance break down into simpler substances. Okay, so this final one is kind of a more broad and generalized definition. So, okay. However, uh, sometimes I also heard a definition from actually I'm teaching fermentation technologies for like 1 year, and I frequently encounter or hear a definition from my students. The define is that like fermentation means changing something, changing something into something in anaerobic condition. That is this definition: any energy releasing metabolic process that takes place only under anaerobic condition. That means produce something using microorganisms in anaerobic conditions. But please keep it in mind, this definition is actually not fully correct. Why? Because the fermentation process, it can happens both in aerobic and anaerobic conditions. I will explain it by an example. Say you want to produce alcohol using yeast. Yeast is a microorganism. Okay, so you want to produce alcohol using yeast. So the yeast will convert your raw materials. So that raw materials can be a carbohydrate source. It can be like, for example, molasses or grapes. So yeast will convert the carbohydrates of these raw materials into alcohol. This chemical change will occur in anaerobic condition. Okay, but say, say you want to produce yeast itself. Basically, actually, the yeast are produced commercially because you have heard about the soda, right? The baking soda. What is actually baking soda? Baking soda is actually yeast, or yeast. You all know that the yeast are used in the baking industry to make bread. So if you want to sell yeast, you need to produce yeast, right, in large amount. This actually done by fermentation process, and if you want to produce yeast, that need to be done in aerobic condition because yeast need oxygen for their growth. Okay, so that means fermentation can be done both in aerobic condition or anaerobic condition depending on your purpose. Okay, so that's why it's good to understand the difference between fermentation and the anaerobic respiration. In this slide and the next slide, actually, you will find the differences between these two, fermentation and anaerobic respiration, in context of like definition, intracellular or extracellular process, oxygen is required or not, what happens after the glycolysis, and what happens in vitro. So I'll just go through. So, fermentation refers to any group of chemical reaction induced by microorganisms to convert any substances to simpler form. Okay, it may happen in presence or absence of oxygen, that is aerobically or anaerobically. Anaerobically. Why? Anaerobic respiration refers to a type of cellular respiration that occurs in the absence of oxygen. Okay, and usually fermentation is an extracellular process, but anaerobic respiration is a intracellular process. I already talked about the requirement of oxygen. And after glycolysis, in case of fermentation, glycolysis does not follow citric acid cycle or electron transport chain, but in anaerobic respiration, glycolysis follows citric acid cycle and electron transport chain. Okay, and in vitro, I mean by fermentation process, if you produce enzymes, you can extract that enzymes from the fermenting cells and use this enzyme in other processes. Okay, but in case of anaerobic respiration, if you extract the enzymes from the cells, you actually cannot use this enzyme to do anaerobic respiration in vitro. In vitro means outside the cell. Okay, so these are the basic differences between this process, fermentation and anaerobic respiration. Okay, so now let's talk about some history. Okay, so this slide actually shows the chronological events in the development of fermentation industry. So I'll not, I mean, read talked very details about this one, and you also don't need to memorize all these things. Just try to have a look. You know, it's good to know the history. History is always fun, and it's actually give us a vision or idea how people used to think. Okay, or what was the chronological events. So okay, so just so I'll not read. I mean, talk much about this one. Please read this by yourself. So you know, we already know. So what fermentation is now? What we understand by fermentation techniques. Okay, so we know we just listen to the definitions of fermentation. So now, what is the definition of fermentation techniques? Okay, so fermentation techniques means the techniques that are used for large scale production of microbial products. Okay, and these techniques must both provide an optimum environment for the microbial synthesis of the desired product and can be economically feasible on a large scale. Actually, it must be economically feasible on a large scale. And the fermentation techniques, they can be divided into two types. One is surface type, another is submersion types. Okay, and among these, in case of surface techniques, the microorganisms are cultivated on the surface of a liquid or solid substrates. Okay, however, this I mean surface techniques, they are kind of complicated, so are very rarely used in industrial setup. Okay, uh, in the contrary, in case of submersion techniques, the microorganisms, they are grown in liquid medium. Okay, and most processes are aerobic, and for this the medium must be vigorously aerated. Okay, basically, you know, for the growth, microorganisms need oxygen. Okay, so if you want to cultivate the microorganisms in the liquid media, you have to provide sufficient air into it. Okay, and this process, I mean, how we provide oxygen into the media is called aeration. Okay, so if you want to cultivate your microorganisms into liquid media, you have to vigorously aerate them, vigorously insert oxygen into the medium. Okay, and usually and actually all the important industrial processes that are industrial process means industrial fermentation processes are carried out by submersion process. Okay, for example, if you want to produce biomass, okay, or protein, antibiotics, enzymes, or if you want to use microorganisms in sewage treatment plant, you have to carry these processes by submersion process. Okay, and the submersion process, okay, you can run this in usually you can run this in three different type of reactors. Okay, one is batch, another is feed batch, and the third one is continuous reactors. Okay, actually there are three types of fermentation. I mean, there are three type of techniques usually used in the industry. Okay, one is batch, another is feed batch, another is continuous reactors. Okay, however, I'll try to talk in more details about these three reactor types in a later part. Okay, so now all the fermentation, I mean important commercially fermentation processes, they can be categorized or grouped into five major groups. Okay, so these are those five major groups. One, the processes which are those are used to produce microbial cells or biomass as the product, microbial enzymes as the product. Number three, microbial metabolites. Number four, process that are used to produce recombinant products. And number five, process that are used to modify a compound which is added to the fermentation. Okay, so it is also called transformation process. I mean, we want to in transformation process we transform some substrate from one substrate to another, or we slightly modify them. Okay, so in the next, I mean from the next slide, I'll talk about these five groups in little bit more details. Okay, so first let's start with the microbial biomass and enzymes. I mean the process by which we produce microbial biomass and enzymes. Okay, however, so the microbial biomass, they can be divided into two major processes. One is production of yeast, which are used in the baking industry, and then number two, production of microbial cells. Actually, nowadays many microbial cells are produced to use them as food, especially animal food, which are called single cell protein. But people are also trying to produce single cell, I mean microorganisms, which can be used as animal food. Okay. Okay, so not only these two, I mean by I mean production of yeast or microbial cells uh, are the major processes. Okay, so there are also processes in which we actually want to produce enzymes. Okay, by using microorganisms, and which enzymes which has commercial values, because in many industries, especially the food industry, feed industry, textile industry, pharmaceutical industry, lots of enzymes are used. Okay, so this huge amount of enzymes are by produced from microorganisms. Okay, so usually the processes by which we produce this thing, yeast or SCP, single cell protein, or microbial enzymes, these processes have some similarities. Okay, because say if you want to produce yeast, you have to grow the yeast. Now think, if you want to produce enzyme, yet you have to actually produce the microorganism itself, because who will produce the enzyme? The microorganism will produce the enzyme. If you can I mean produce the cultivate the microorganisms, they will automatically produce the enzymes. Okay, so that's why the processes are kind of similar if you want to produce whether the biomass or the enzymes. Okay, this slide it shows some examples of commercially important enzymes. So you don't have to memorize all these things. So I mean in this table you will find the name of the enzymes and their application, in which industry they are used, and the source of the enzyme, such as for example the first one, amylase. This is actually a very important enzyme. Okay, it is used in baking and milling industry. So in what purpose? The application is to reduce the viscosity of the dough. Okay, uh, and acceleration of fermentation, increase in low volume, etc., etc. and amylase is usually produced from fungal, definitely using fermentation process. Okay, so there are also many other commercially important enzymes here. You don't have to memorize all these things. Okay, just go through this one. And this slide, it uh, actually kind of, you can say, a small summary of the this slide. Okay, so so these are basically same, so you don't have to read this in details. So let's move to the next slide. This slide shows some commercially important acids which are also produced from microorganisms. Okay, so this the leftmost column, it shows the name of those commercially valuable products, such as, for example, acetic acid. It is produced by acetobacter, and the substrate for acetic acid is sugarcane juice. Citric acid is produced by Aspergillus niger, and the substrate that we use to produce citric acid is beet molasses. Okay, so I have already talked about the process which are used to produce microbial biomass and enzymes. Now, microbial metabolites. Okay, so microbial metabolites are two types. One is primary metabolites, and another is uh, secondary metabolites, and both are important. So, which are primary metabolites? Such as amino acids, nucleotides, proteins, nucleic acids, lipids, carbohydrates, etc. These are called primary metabolites because these metabolites is needed by the microorganisms itself for their growth. Okay, and that's why the primary metabolites are produced by the microorganisms mainly in the log phase of their growth. Okay, in contrary, the secondary metabolites, okay, they are not required for the growth of the microorganisms. Rather, these secondary metabolites are required for their survivability. Okay, so that's why usually the microorganisms, they produce secondary metabolites during their deceleration or stationary phase of growth. Okay, because I think you all know the microbial growth curve, right? They have the the lag phase, log phase, stationary phase, and the deceleration or death phase. Okay, so basically, when the microbes are in the log phase, they grow rapidly. So that's why they need those kind of metabolites which are important for their growth. But when they're at the, when the microbes are at the stationary phase, at that phase, it's difficult for them to survive because the nutrients get depleted, the conditions get tough. So they need at that time, when they're at the stationary phase, the microbes need those metabolites which are important for the survivability. Okay, so example of secondary metabolites is antibiotics and toxins. Okay, so anyway, these information are actually very important because suppose you want to produce antibiotics, so you have to design your fermentation process in a way that your microbes will be in the stationary phase for a long time, right? So if your microbes are in the stationary phase for a long time, you will get more secondary metabolites. But if you want to produce primary metabolites, so for example, you want to produce amino acids, okay, so in that case, basically, you have to, your design of the process have to be such as the log phase, okay, or which is also called exponentially growth phase. It should be longer, because if you can make this step longer, you will get more primary metabolites. Okay. Anyway, so the next slide, this slide, this shows some example of primary metabolites which have commercial significance, such as ethanol. Okay, we all know why ethanol is very important. So citric acid, glutamic acid, lysine. Okay, lysine is used as a feed supplement, nucleotides, flavor enhancers, vitamins, feed supplements. Okay. So now the another process by which we produce recombinant products. Okay, and products produced by engineered microorganisms include insulin, interferon, human serum albumin, factors 8 and epidermal growth factors, calf chymosin, bovine somatostatin, etc. etc. However, when you want to, when your goal is to design a process by which you will produce recombinant products, some factors should be kept in our mind. Okay, so what are those factors? So, for example, by using genetic engineering, you modified your microorganisms which will produce your target products. So, it's important that this target product is secretory. That means your genetically modified organisms will produce that product and will secrete it outside their body. So it's important because if the product is secretive type, it's easy to harvest that product after the fermentation. Okay, so this is an important factor. Another factor is minimization of the degradation of the product. Okay, so during the process your product will be produced, and it's important that your product will not get degraded during the process. Okay, and and you should be able to control the synthesis of your product during the fermentation. That means you should have the ability to control the fermentation process. And another fact is maximizing the expression of the foreign gene. What that mean? Suppose, say you want to produce insulin, okay, by using bacteria. So that's why you have genetically modified your bacteria. That means you inserted the insulin producing gene into your bacteria. Okay, so now in the fermentation process, when the bacteria will grow, if they have the gene, but if that gene is not expressed, insulin will not be produced, right? So if your bacteria don't express the gene, or express the gene not very vigorously, in a minimum level, then your product will be also minimum, right? But if your modified organisms express the highly express the gene, you will get lots of product. Okay, so this is important. Whenever you try to genetically engineer your organisms, it's important that your organisms will express the gene. Okay, will highly express the gene. Okay, so that was all about the recombinant products. Now then let's talk about the transformation process. Okay, so in transformation process, basically what we do, by the name we can guess, we transform a substrate from one form to another form. Okay, so in this case, we can do this very efficiently by using microorganisms. Why? Because microorganisms can behave as catalyst with high specificity. Okay, microorganisms are usually very specific. Okay, and microbial processes are more specific than chemical ones. They are more specific than the chemical ones, and enable the addition, modification of functional groups at a specific position on a complex molecule without the use of chemical protection. That means, suppose you want to add a particular functional group, suppose OH group, okay, into your substrate. Microorganisms can do it very efficiently, more efficiently than the chemical ones. Okay, and also one good part of using microorganisms is that usually this process, you can do this process in low temperature and pressure. So if you can do your transformation process in low temperature and pressure, it means actually it will require less energy, right? So if less energy is required, your costing will be less. Okay, so we are done with the the major fermentation processes. So now I'll talk about the fermentation process itself. Okay, so although there are different fermentation process as we just talked. However, all the fermentation process or the typical fermentation process, basically they have 6 basic components. Okay, so all the fermentation process basically they have 6 major components. So what are the components? First, formulation of the media. Okay, you have to formulate the media which is optimum for the growth of your microbes. Okay, and then sterilization of the medium, fermenters, and ancillary equipments. Okay, so before you start your process, run your process, you have to sterile all the things. You have to sterile the media that you have just formulated and prepared. Okay, you have to sterile the fermenters, or which is called bioreactors itself, and all the other equipments that will be used during the process. Okay, the third component part is production of an active pure culture in sufficient quantity to inoculate the production vessel. Production vessel means the fermenters. In the fermenters, you will put your raw materials and the your microorganisms. Okay, so this is actually called culture. Okay, so before going to the main fermenter, you actually have to produce sufficient amount of your microorganisms. Okay, which is called pure culture. Okay, and this pure culture should be in its active form. So if you can put active pure culture into the fermenter, so then after you put those active culture into the fermenter, the process will start rapidly. Okay, so the fourth part, okay, now the fourth component part is the growth of the organism in the production fermenter under optimum conditions for product formation. I mean, inside the fermenter, you have to provide the optimum conditions. Optimum condition means optimum pressure, optimum oxygen, optimum temperature, optimum pH, optimum say sugar level, or all the other chemicals necessary. Okay, and the fifth component part, the extraction of the product and its purification. Okay, and the sixth, the disposal of effluents produced by the processes. Okay, so these 6 are the major or the basic component parts of a typical fermentation process. Okay, so that's all for this lecture. In the next lecture, we'll talk about the steps of the fermentation process. Okay, so please don't get confused. These are the component parts of the fermentation process, but all these component parts basically they fall into 2 steps. One is upstream stage processing, upstream, another is downstream. They are called upstream processing and downstream processing. Okay, so in the next lecture I'll talk about this upstream processing and downstream processing. Okay, till then, be safe. Bye bye.